episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show with another really fascinating guest for you today, involved in creating a better tomorrow on many different fronts. Today, we have the honor of being joined by Dr. Veronica RodriguezBravo, who is assistant professor in the Department of Cancer Biology here in Philadelphia at the Sidney Kimmel Cancer Center at Jefferson University. Dr. RodriguezBravo obtained her PhD in Pathology and Cell Biology, Summa Cum Laude, from the University of Barcelona back in 2007, where she received the Extraordinary Doctorate Award for her studies on the distinct DNA replication checkpoint mechanism as tumor cells. During her postdoc work at the Experimental Oncology Department at the University Medical Center of Utrecht in the Netherlands, and at the Molecular and Cell Biology Program's Memorial Sloan Kettering Cancer Center, she specialized in the study of chromosome segregation during mitosis and the role of nuclear pores in genome integrity maintenance, and we'll be getting into that topic a little further. Her postdoc work allowed her to apply genome editing techniques crucial to dissect the various functions of mitotic and nuclear pore proteins in chromosomal stability. It resulted in the recognition with Memorial Sloan Kettering Cancer Center Postdoc Research Award. And her lab's current focus is on the study of genome integrity mechanisms and the relationships of the defects in cell division to cancer pathogenesis, with special emphasis on pathways that contribute to cancer cells and these more aggressive phenotypes. So with that introduction, welcome Dr. Veronica RodriguezBravo to the show.
Thank you so much for the nice and warm welcome. It's a pleasure for me to be here with you today, and I hope we have a great time discussing about science, and we have something interesting for everybody to follow.
I definitely think we will. There's a lot of really interesting and new topics here that I'm really looking forward to getting into with you. But you know, I love to start off just by handing our guests the floor for a little bit to talk a little more about themselves. Veronica, you could sort of take us a little back to the early days, everything from where you grew up, how you got interested in science and in medicine and oncology, and sort of the early days of your research. I think that'd be a great way to lay the ground for what we're going to be talking about.
Sure. So I was born and raised in Barcelona, Spain. So that's where I did my initial training. I went to college, and initially I always had a big drive for science, but I also like other things. I always like to point out that to young people starting, like they may not know what they want to do, and that's fine. You may have some doubt. You want to try this, try that. So I didn't know I was going to be a scientist. I just like science. And I was one of those kids who asked for a microscope for Christmas when I was, I don't know, maybe 7-year-old or something like that. But I also like art and, books, painting, music. So I was a little bit like, I don't know if I want to commit only to one thing. But at some point I decided to go ahead and go study biology. I figured that I couldn't learn by myself to work in a laboratory. I could play guitar at home, I could paint at home, but you cannot run a lab at home. So I thought, okay, let's go to college, study the fundamental principles of biology, cell biology. And then I had a very strong drive already from year one. I wanted to study cancer. And that probably was related to back in the time, and it's currently the same, but back in the days, it was even more dark. The knowledge about cancer, why people were getting sick around us, family, friends, it was scary. And I had that feeling of scary knowing about cases of people getting sick, right? But I had questions. So I was thinking, why? How is this happening? And so I wanted to learn more at the fundamental level. How are the cells in our body turning into this? What is the biology behind that? So I started early in my career thinking about these things, connecting to teachers, professors, to try to get more lab involvement to get to that point. So when I finished college, I decided I had a very good idea that I wanted to do a PhD to interrogate more of these things, learn more, and become a scientist. So learn the business, let's say. And I didn't know much when I started. So I just went step by step, learning from, my mentors, my colleagues. And I realized that I wanted to learn more, and I wanted to go farther, and I wanted to get more experience. And after my PhD, I wanted to get different backgrounds and different information. Usually you get more techniques. The more experience you get, the more techniques you learn, the more you can apply to answer questions. So I knew I wanted to learn some of these new techniques abroad. So I went first to the Netherlands. It was closer to Barcelona, so there was an easier choice for a young scientist to make. And then I moved to the United States in 2008, and I was lucky enough to go to one of the main places in the world to study cancer. And this Memorial Sloan Kettering in New York. So, and there I had my heavy training, which I learned a lot, and with my mentor there. And I learned to manipulate the genome of cancer cells and to get answers. So we manipulated cells genetically so we can delete things at will. And when I say delete, I mean to get rid of a gene. Sure. Like with a click, let's say, and then see what happens and analyze what we call separation of functions. So we want to understand the complexity of biology. Sometimes it's that proteins, sometimes they don't do only one thing. They may be doing several things. So when you get rid of them, you cannot separate all these different functions. Everything explodes kind of, right? So we had to do some genetic tools to separate different functions of a protein that is very important for maintaining the stability of the genome when cells divide. Yep. What is called the mitotic mitosis and mitotic checkpoint that allows cells to, every time they divide, they don't lose chromosomes or they don't break them. And so we studied those pathways, and we found something interesting, which is that there was a connection to of these proteins that many other people were studying with their specific location in the periphery of the nucleus of the cell. And those places were nuclear pore complexes, which virtually are holes. There were holes in the nucleus, so it's kind of like a tunnel through which the cell communicates the nucleus and the outside of the nucleus, and they transport things through those holes. But they are more than transport, they are more than tunnels, and they seem to be regulating many other things. And one of the things we found is that they can regulate the stability of the genome as well through these regulatory networks. So that was kind of a novel finding and unexpected and very interesting, because then it opened a new door for me and for others in the field to investigate these new functions of proteins living there in those, what we thought were only holes, but they are not only holes in the nucleus. And they are modulating many other functions. So as a matter of fact, we are studying chromosome segregation and chromosome instability, but we're also studying how proteins in the nuclear pore regulate the gene expression. Yep. And that means how genes are turned on and off. And that's very important for normal cell, but also very, very important from the perspective of disease and cancer. So we know now that many cancers have huge changes in the way they turn on and off genes. That's a way that cancer cells use to their advantage. So they really select things they want to be turned on all the time. The chromatin regulation is what we say, chromatin regulation, transcription, all has to do with how cells can modulate how they express the genes to make proteins and to gain functions. And that leads to more aggressive tumors. So nuclear pore proteins can regulate all these things too. And those are also the things we are looking at the lab. So the more we investigate these new mechanisms, the more we are finding that they're touching different aspects of the cancer cell. And they may be selected for that reason, because they promote survival of cancer cells to threats, to stress, to, for example, therapy when they're treated in tumors. But they can also change the way the genes are modulated. Yeah. And that's a very powerful tool for a cancer cell, because they can change that at will, depending on, and that's important when cells become very aggressive and you treat tumors with different therapies. There are always resistances, right? Sure. The tumors can come back with a particular resistance, and you treat with treatment B, and then they become resistant. And many in our field and in other fields think that that is very related to the way cells can modulate chromatin. And they can adapt to whatever you throw in them and change the way they turn on and off these genes. So we think the nuclear pore is also playing an important role in there. That's something that wasn't known years ago. So we think it's an interesting, novel approach that may give us some answers. And so new answers to all questions that still are met needs from clinical perspective. So that's where we are trying to put our efforts.
Very, very exciting. It's very exciting, and you said a lot. You said a lot of very, very interesting things there, but a couple that really resonate. One, sort of this concept that whether we're talking about a gene or a protein, they don't just do one thing. They can do a lot of different things depending how they fold and spliced and so forth and so on. And you know this better than I do. And then, when we, I love referring to so-called, the so-called architecture of sort of the gene expression in the cell. We think, obviously, we spent the last couple of decades a lot on the genome, the last decade or so talking about the epigenome and sort of the non-gene changes. And now you're bringing in a couple other really interesting angles that we don't hear as much about in terms of this topic of the nuclear pore complex. And I did a little sort of background before coming on the call with you. So we have something like 1,000 of these nuclear pore complexes per cell, about 110 megadalton structure, and a couple hundred individual protein molecules that make up these structures. So obviously, I know you lecture on this a lot all the time, but can you take a couple of minutes and just talk a little bit more about what we're talking about here? Because this is, once again, it's not, they're holes, that's great, but they're really a very complex structure here that is sort of modulating what's going on in the nucleus and then a lot of the stuff in the cellular milieu, let's say. Talk a little bit more about the NPCs, if you will.
Right. So the nuclear pore complex actually has been studied from the basic perspective, from biochemistry side for decades. Yeah. So people, obviously from early on, we knew that the way the cell is organized, we know it has a cytosol, which is kind of in the external part of the cell and with a membrane. And that's the first thing that when cells are in contact with the environment, that's the first thing that molecules encounter. But then in the nucleus is the most preserved part, and it has an envelope around it. So it cannot be exposed because we want to protect the genome. All the DNA is packed in there. Yeah. And that's the genome that has to be making proteins, but also that is partitioned when cells divide. But all the time is covered by this nuclear envelope, but there has to be some communication. So we have to imagine that this is like a little ball without another ball, if we think about it, right? Like almost like a chocolate. You know those chocolates that have like an inner part. There is a different component from the outside. So
I love those.
Part, so that's so tasty. So in the nucleus is where the tasty thing lives, right? Is the main code that controls everything else. But it can be damaged easily. So it has to be extremely well protected there. But it needs to be communicated. So if we think about it also like a golf ball almost that has little indents in it. So all around it, it has these holes. You said like 1,000, that would be like an average number in a cell. That's correct. Numbers can change. So we found that in aggressive tumors, we detected more increase in those pores than in primary tumors, for example. So cancer cells even can create more of those pores to potentiate things like transport of molecules so they can modulate better what they want in the nucleus. So at the end of the day, you have like a 3D, round nuclei all covered with little holes around it. And there is continuous transfer of things through them. So every single protein that has to go to a nucleus that is bigger than a certain size have to go through those holes in a regulated manner. And then when the genes are turned on, they create these RNA sequences that now people are more familiar with RNA based on the vaccines, right? Sure. So that's the first, let's say like a code that is made from the DNA of the genes, and that has to be exported outside to make the proteins. So there has to be this continuous circuit in which proteins go inside, transcription factors. These are the ones who bind to genes, activate the gene to make this transcript mRNA. The mRNA has to go outside through the pore and be transcribed into protein. So that has to be constantly moving. Things are constantly moving through these pores. So they are fundamental to the normal cell homeostasis. So they have been studied for many, many years from basic laboratories. So they figure the structure of this thing, how it's assembled. It's a big megadaltonic structure with many different copies of each individual component. And it has to make a particular shape that has to be preserved. Otherwise, the transfer wouldn't happen efficiently. And so we know how it looks. We know what proteins live in the part outside of the nucleus, which proteins live in the inside. And what is missing right now is the functionality. What things are interacting with what? Why this is happening in one direction and not in the other? And what is this regulated in cancer or in disease? So the fundamental structure we kind of understand. Why these things are deregulated in cancer, that was one of the things that took me into the field of cancer biology and nuclear pores, is because we didn't know really. We knew because there has been a lot of sequencing of the genome, right, in the last 20 years or more. We know about a lot of mutations that are found in cancer, and a lot about why some things are super up why some genes are very upregulated in cancer versus others. So we know, as we published, that in some of the tumors we studied, like prostate cancer, in very aggressive tumors, they have an increase and a change in these proteins of the nuclear pore. Yep. And we question, is that important? Is that contributing in some way to the aggressiveness of those tumors? Because there was nobody had looked at that. And many times we have this genome-wide information, but we still don't know what every single thing is doing, right? Mm-hmm. So that's where we wanted to understand, okay, these dysregulated nuclear pore components, are they doing something? Are they contributing mechanistically to the aggressiveness of the tumors? Are they providing more advantage for the growth of the tumors to resist therapy, things like that? So that's how we started interrogating. We know this is different in tumors versus normal. Mm-hmm. What does that mean? So we went and dissect every single difference that we observe in analyzing these databases from patients, actually. Mm-hmm. And we started dissecting one by one these components of the nuclear pore, and we're starting to find out what each of them is doing. Mm-hmm. And we started with one that we found it regulates nuclear transport. And we found that it actually makes these transcription factors to go faster into the nucleus and activate more genes. Mm-hmm. And that's something that aggressive tumors like, because they have their favorite pathways and mechanisms that are regulated, that if they can turn it on more and more, they grow faster and more aggressive. So we defined that. And now we're also interrogating how this also is linked to increasing chromosomal stability. Yep. And it's important to contextualize that in cancer, we know there are aberrations in the genome. So there are mutations, there are defects on chromosome content that accumulate during the course of the disease. And the more aggressive and advanced is a tumor, the more of these alterations accumulate. But there is a paradox. And there is a paradox in the chromosomal instability and aneuploidy field, which is we know that there is a certain level of these aberrations that are not well tolerated. Normal cells don't like it, but tumor cells don't like it either. Yeah. So how they do it, how they continue accumulating all of these aberrations, because they really accumulate a lot of defects. They have aberrant nuclei shapes, aberrant chromosome contents. They have pieces of DNA floating here and there, but they're still alive. So there is this paradox of how they compensate for that. How can tumor cells survive to this aberration? That's something that we are investigating, and we think the nuclear pore complex is also involved in that. So we're trying to define what is there, and if we can find new vulnerabilities that have to be contemplated before, because most of the time we're looking always at the same players, right? Androgen receptor is very important in prostate cancer. P53 is one of the canonical tumor suppressors that is always mutated in almost every cancer. But we need new answers, new players to complete the picture, and maybe find new therapeutic angles, and that's what we are going for. We want to understand new pieces of biology that may be important to find specific therapeutic opportunities to target tumors that may not respond to other things. That's where we are trying to dig in.
Excellent. And thinking about these aggressive. I wasn't had a chance to go through some of an extensive list of publications, but the couple that popped out at me, so a little bit dive into here based on what you were talking about. There was in 2020, Nature of Urology, in May, you published the cellular rewiring in lethal prostate cancer, the architecture of drug resistance, and then a related paper to that in 2018 in Cell, nuclear pores promote lethal prostate cancer, increasing POM121, and then a lot of the transcription factors listed there. And one of the very interesting things here, as you're mentioning, there's obviously the common things that a lot of folks target, but then you're highlighting in both these papers, not just some of, for instance, the prostate cancer specific genes and so forth that make those tumors more aggressive, but then very interestingly, this reprogramming to more of an embryonic state, which, this fascinating connection to, we hear about a lot of cellular reprogramming in the stem cell space, but also the ability of now cancer cells to use some of that same knowledge to differentiate themselves and explore, let's say, cancer sort of state space, sort of the systems biology folks like to use those words. Take us a little bit, because as you're saying, these nuclear pores trickle down to so much, whether they're cancer specific or stem cell type phenotypes. Take us on a little bit of what you're discovering there in terms of sort of this combinatorial basket of the way these cancer cells use these changes to escape. And then on top of that, one of the very interesting parts in the 2020 paper that you mentioned is that, you know, understanding these changes is going to require new types of therapeutic regimens that may involve combinations, as opposed to sort of the more traditional thing, where you think of these smart drugs nowadays. When you got so much going on, you know, you need more than one drug. Talk a little bit about your concepts with regard to combination therapeutic regimens for some of these changes.
Yeah, so you touch upon a very interesting concept that it's actually, to me, and when I discuss this with colleagues, kind of the jewel of the crown of the cancer biology field right now, is the what you mentioned, the cancer stem phenotypes. So that has been discussed for many years.
Yep.
But I think it's more open concept now, more accepted, that many tumors, and that's true for prostate, but also for other tumors, specifically when they are exposed to therapy, they have the ability to transform into something more undifferentiated. And they start acquiring characteristics that remind more to a stem-like, to undifferentiated developmental pathways. These all remind us that those cells are not like terminal differentiated anymore. So they are more plastic, and they can go into a more less defined tissue-specific phenotype. So that means they don't look like prostate anymore. Sometimes they can look similar to like a small cell lung cancer or neuroendocrine tumors. They share features that they even seem that they don't come from the origin of the tumor, and those become very aggressive. And it's a very interesting. Now it's a very hot topic, actually, and we think that's a very important thing, because there's a way of thought in the field, in prostate and in other tumors, that those cells may exist already.
Okay.
They don't originate because you treat the tumors. They may be already there, and therefore they are the ones that are able to adapt and to survive therapies, and then give rise to a progeny of other cells that can differentiate into different lineages. So those could be the ones that we should target, but it's how we target them. That's the big question. And also they are become more abundant after therapy. They may be there, but after treatments are applied, they are getting rich. And we have seen that in the past in some of studies that undifferentiated subpopulation of tumor cells, those are the ones that are enriching, for example, when in chemotherapy resistant populations. And then they differentiate again. So they have this plasticity in which they can kind of do different cell divisions depending on if they want to look the same and remain the same status, or they want to differentiate into daughter cells that may look less stem. So they have this capability. So the question is, what are the pathways that can be targeted there? And that's where a lot of people are trying to understand if epigenetic therapy will work for those. What are the pathways these are dependent on? And one of the questions we're trying to answer as well is, there are differences in nuclear pores between these undifferentiated cancer stem cell populations and more differentiated cells. That could be an interesting question that, to my knowledge, nobody has really addressed, and we will be in the position to actually find out, just comparing, because we can't separate those populations from real tumors or from in vitro studies. We can separate them and study them. So that's something we're starting to do, to look at the nuclear pore and if there are differences. Because those will be novel approaches to try to tackle how we actually attack the mother, right? The mother cell of the tumor. The other thing that is very interesting added to that is the combination therapy. And the more we're learning is that probably we're leading into the more the better. So combining different strategies into one single treatment. And the idea will be specific for a patient, because whatever is working for patient one may not work for patient two. So it will depend on if we have the information of the genetic mutations a particular patient has in a particular tumor. But beyond mutation, I think we need more information, because many times the mutation is not going to give you many things that are mutated are losing their function. So you cannot. Therapies are oriented to inhibit something, not to activate something. Most of the therapy. That's the problem with the tumor suppressors. We need gene therapy to activate them. And p53, for example, is a tumor suppressor. So we probably will have a huge tool if we could activate p53 in tumors. But so far, the therapies go the other way. So we try to inhibit whatever it's an oncogene, or that it's giving an advantage to the tumor and it's upregulated or gaining a function. So that's how therapies work. So we need more than mutational landscape. We need to know what is turned on. So that's why we need RNA sequencing and transcriptome data. And now people are starting to do single cell analysis, which is very powerful. So you get the whole tumor and you get the information of each individual cell instead of a mixture of everything. So you know the populations that are there. So we can separate populations. And there is where you can go to see, okay, show me the populations that have the markers of stem cells, and show me the populations that have the markers of a differentiated cell, and show me what transcription factors are upregulated or downregulated in each of them, and now try to tease apart what population could be targeted with what. Mm-hmm. And I think the chemotherapy, many people may be against chemotherapy, but chemotherapy is still widely used, and it works. I mean, it's the approach that we have. It's not specific, but sometimes we have to wonder what is better: general therapy that kills a lot of cells, or very specific therapy that can create very quick persistences. And we have had this discussion like 2 weeks ago in a prostate cancer meeting that we have, a conference, in which that was one of the questions. From a basic lab perspective, what should we prioritize? Like more systemic general therapies that we know may affect multiple pathways, multiple kind of cells, lineages, may have more off-target effects, but they are efficient and they can really reduce the tumor quick. Or we should go very specific to a one protein, one target, or we should combine both. And the feeling is that probably we're going to be combining more than one or two or three in the future, obviously considering always that the toxicities and the side effects.
Sure.
But taking advantage of the vulnerabilities of particular tumors, and maybe chemotherapy plus in prostate cancer, we're seeing that now they're earlier in the disease stage in the tumors, they're adding chemotherapy to anti-androgen therapy. Mm-hmm. Whereas in the past, chemotherapy was only added later on. And now they're seeing there are benefits combining early on in the disease. So it seems that the sooner and the stronger you hit, the better. So maybe the future is going to be, we're going to go very hard and hit very hard early on with everything we got. And the more we know about the tumor, maybe will allow us to utilize all the different drugs that are available that may be used for other tumors that are maybe still in clinical trials, but offer that to people. Your tumor potentially could respond very well to this new drug in combination with what we know. So I think in the future, we may be in that scenario in which we will be able to utilize more of one or two or three drugs if they can be combined together or sequentially. The earlier the better, because you develop less resistances than later in the tumor progression. We're not there yet, but we all agree that we may be getting more and more into that space, and at least we'll have more options. And crucially is which patients could respond to what, because we don't want to give something to patients that may not respond, and we should have known that based on their genetic information of the tumor or the gene expression profile of the tumor, things like that. So something like that should be integrated in the future, so clinicians can have access to that information. What is the pipeline of drugs that have been tested, and how to capitalize on that for specific tumors, for specific patients. And I think that will be the future. And hopefully we can get there better and better. There is very exciting things coming out, at least from the scientific side, not from the main public. And we are seeing huge game changers in clinical trials now for many different tumors, and there is optimism. And then there is the immunotherapy side, of course,
Sure.
that the goal is to make it work for many tumors, not only for some.
Sure.
That could combine, that usually is, the goal is to combine immunotherapy with all these other things. Mm-hmm. The idea is that some treatments may turn tumors more reactive to immunotherapy. Yep. So the combination could be also combined with immunotherapy and make a perfect combo, ideally in the future, to kill tumor cells.
Yeah, it's a fascinating time in terms of both what you're doing and in sort of looking at these lesser sort of explored targets, and at the same time understanding that we have all sorts of modalities that we can combine. So it's really interesting. But then I have to go now to the flip side of all this, because you're focusing on amazingly interesting targets. You're also using some of these cutting edge tools in terms of the CRISPRs and the single cell analyses and so forth, personalizing everything. But then you have a last month you published this fascinating review, Cancer Treatment Reviews, June 2021, entitled, Redefining Cancer of Unknown Primary, Is Precision Medicine Really Shifting the Paradigm? And you introduced us in this paper to this topic of cancer of the unknown primary, defined as the absence of a clinically identified primary lesion, where the primary is not seen at the time of diagnosis despite standardized diagnostic workup. And then you present this fascinating info that 2 to 5% of all cancers are defined as cancer of unknown primary, and up to 15% of all new cancers are presenting a so-called malignancy of unknown origin. And this is just, for 2021, this is I've never heard this term before. I mean, because I'm not in the oncology space, but talk a little bit about this, because we see so much in the sort of the popular press about, oh, this gene and that type of cancer, but only 50% of all cancer, we don't know where they're coming from. Balance that one out for us, because I think this is fascinating.
I mean, we all like to classify, right? So we work with classifications, even statistically. When we talk about tumor types, the incidence, the number of deaths per year, we go to the classical statistic, and is separated by gender, if you want, and the tumor types, right? But because we know the tumor type, right? We know that it was a prostate cancer, or a lung cancer, or a breast cancer, and this is the number of people that got it. But then there is a percentage of tumors that people arrive to the clinic with a disseminated disease or a nodule here or there that is a metastasis, and they don't know where they came from. And that's a reality that many clinicians confront. So those are the ones that are called like the primary metastasis of unknown primary, because they couldn't figure out exactly with 100% confidence what was the tumor, the tissue that originated that tumor. And that creates a challenge, because in many of these tumors, for example, breast cancer or prostate cancer, based on their expression of certain genes, for example, in prostate cancer, if they express androgen receptor, it came from the prostate. Yeah. But if you cannot even detect that, you don't know where it came from, because some tumors of prostate originated from the prostate may lose androgen receptor. Mm-hmm. So the thing is that these tumors that are metastatic, because they know they don't belong to the tissue where they are, but they don't find the origin, they're still not understanding on if the primary disappeared, or if it's too small to detect, or if it regressed. And the only thing that they know is that they detect this tumor, they try their best to find out where it came from, to identify the best therapy they could apply. Because if it's coming from the breast, it may respond to, they may look at the typical markers for breast tumors and the different classifications of what therapies they could apply. But that involves time, and usually those tumors are very aggressive. So they're typically more aggressive than the ones that are presented when the primary. And one of the particularities they have, and that's why basically I got involved in this review, is because they tend to have higher levels of chromosomal instability than other metastases that are known where they come from, which is kind of fascinating. And that could be an entry point for therapeutic angle as well, because they may be more vulnerable to certain things. So that's how we started discussing, and there were some studies actually describing that these tumors, they're trying to find out how to define them, right? And where are they coming from? So they did epigenetic screenings to see if there were differences. And one of the main things that stood out was the aberrations in chromosomal content. And basically, it's still an unknown. That's something that the clinicians have to confront, and they try the best they can therapeutically, but usually they don't go well, because they don't know what they have in their hands. And it's a very good question, where are they? And if you cannot identify, many times the primary is identified in the autopsy, and sometimes not even then. So the question is, what's going on in here? So these may be very aggressive tumors that early on disseminate, even before the primary tumor gets detected. And those typically, when tumors do that, they have very, very, very bad behavior. Because many times you have tumors that are detectable, and then you look if the oncologists look if they move into the nodules, lymph nodules that are nearby, and then they evaluate if the nodules are positive, maybe there is some metastasis somewhere else. But there is a size, right, a mass that you detect in the primary location. In this case, these tumors went really fast. If you cannot even detect the primary, the mechanisms for how they got there and how bad they got, you are behind the tumor already. So behind that they are very aggressive, very difficult to treat, and the prognosis is really bad. So the question is how you treat those tumors. So it would be interesting to explore those differences in the chromosomal instability, because, for example, one of the things we are trying to find in my lab is to exploit that. So you have more chromosomal instability, fine. We're going to use that to our advantage, because there must be a way to turn this around and exploit that, so cancer cells cannot tolerate those errors anymore. So that's what we're trying to do. So the question is, once we find those therapies that exploit chromosomal instability, could this be applied to those tumors, for example, regardless of what they're coming from? If they are dependent on their chromosomal instability level, we may be able to target them specifically with certain therapies for tumors that are high chromosomally unstable. Yeah, but it's a fascinating field. When I got into that, I also was surprised I didn't hear or learn more about that before, because as you mentioned, we learn about all these tumors or these mutations or these proteins, but then I started reading about this. This is fascinating, how we are not studying everybody. We should be looking at this, right? Because they usually labs are very oriented into specific tumors. So to narrow down your search and don't spread too thin, some people start breast cancer, some other people are especially in pancreatic cancer, right? Maybe we should also be looking at this type of tumors in collaboration with pathologists and oncologists whenever they have this type of tumors, to try to identify in the labs within that cancer center, for example, can we identify something from this and maybe put some common efforts, because that's a problem that it's everywhere, right? But nobody can understand it well, because I don't know how many basic labs are studying this type of tumors that have an unknown primary. I don't know anybody from a research lab perspective. I don't know any lab focusing only on that. So maybe we should have a common goal for that. Like,
Probably.
we all have different knowledges for different tumors. Whenever this appears, say, okay, we're going to work together to identify what are the markers, because we are all familiar with the markers that we know, right? But I'm not familiar with the markers in pancreatic cancer. I'm familiar with the markers in the tumors that we know, right? So maybe common efforts should be applied to identify specific markers, specific mechanisms, and things like that. Because to my knowledge, people are not focusing on them. And that's why it's still a big question. Even in the title of the paper, there was a question, right?
Yeah.
It's like a question mark. Yeah.
The funny thing, when I was looking at it, the thing that came in the back of my mind, it was sort of that basket of papers that's sitting over here on things like spontaneous remission, which is another thing. It's like, all right, well, this is odd. Let's put it over here. And it got me thinking, like, are these spontaneous remissions of the primary tumor that everything else proceeds? But obviously, there's a lot of exploration.
Yeah, one theory that can be applied, and that there's people working on tumor evolution.
Yeah.
Chromosomal instability is directly linked to rapid chromosomal changes, adaptation, and tumor evolution. So people think that quick errors that are accumulated in a genome allow the tumor population to adapt quickly and change very quickly as well. And if you apply therapy, even more, because you apply pressure. This is like, in the evolution of the species, it's the same thing. So it's possible, one possibility could be that in that primary tumors, if they have very high chromosomal instability, there may be a lot of changes going on early on. Some of these cells that accumulated many changes may be able now to leave, but the original ones that stay, maybe they were not so aggressive anymore, maybe they regress, or they just sit there and they are not detected because maybe there's not enough volume to be detected. That could be one possibility. The other possibility is that they are in a dormant state. I don't know if you're familiar with this dormancy theory. Sure. And more and more people are starting to describe that in several tumors, there are cells that can very early on disseminate from the primary tumor, sit in some other distant places for years, and suddenly in a dormant state, and suddenly awake years later after even the primary was detected or treated. So those cells are able to somehow hide and being like in a standby mode, and being detected by the immune system. And at some point they reactivate, and they don't proliferate because they know to be out of the cell cycle, which is what you need to actually divide. And they have markers of being arrested, not divided. But at some point they reactivate themselves. So it's an emerging field, not so emerging anymore. There's big labs working on this right now. That they really think that can be a cause of these cancers that appear 20 years later. The person was treated, everything was fine, and then something reappears. And and they think that these cells can actually start traveling around very early on and sitting in different places and waiting for the right moment, maybe to accumulate more mutations, or when the immune system, you get older, the host, if we think about the human as a host and the tumors are waiting there, gets older, so it's more vulnerable, the tissues change, and the immune system is weaker, and then it's the opportunity. So there's really people looking at that. And so the evolution concept may be one thing. The other thing is the dormancy concept, which is scary because you cannot detect those things.
That's a really scary topic. Just thinking about it.
The detection, the problem here is the detection, which is one thing that is also improving in the clinical side. And they're trying to work really hard in radiology and this all radiomics era in which they're applying detection with treatment. So you want to see the smaller things sooner and even treat it there and see it with things that are radioactive and you can detect in a scan. Yeah. So the smaller you can see, the better. But those dormant cells, I mean, it's almost impossible to detect. I don't know how that could be detected. So it's kind of a scary concept, but it's a real thing that people are studying. So that could be behind those type of tumors, for example, that the primary cannot be detected because there's not a real mass. It's just a small population of cells that maybe just sit there after some of them disseminated. So maybe this dormancy is heavy in the metastasis of a non-primary, and fuel more with the chromosomal instability they carry. They can adapt and change quickly. So those things could be, but I don't know anybody studying this, and I think it's a fascinating area of study and obviously a clinical problem too. Yeah, but I'm glad you mentioned that because I'm still puzzled about all what I learned when we were writing this review. It's like nobody's looking at this. It's like there are many questions here to be answered, and we need a global approach with different expertise to tackle this, because there has to be an explanation. There has to be, but we don't know.
Well, it's a scary topic, but I appreciate you publishing about it. Even that's the early days in understanding it. I had never heard anything about it, so I was glad to see that you recently are bringing the topic to the fore through these publications. Veronica, obviously, you're focused on cancer, and will be focused on it for a while now. But obviously, these nuclear pore complexes and their ability to maintain and modulate what goes on the genome must affect many other sort of therapeutic area. Are you collaborating with other people that are sort of interested in the NPCs and I off the top of my head, I've seen some things in the press about them, but other disease areas, other therapeutic niches where nuclear pore complex biology is becoming an interesting target?
So in cancer is becoming a very interesting target, but we need to know more beyond the transport function. And then in neurodegenerative diseases, the nuclear pores are important, and that has been known also for a while. The problem there is different. The problem there is sometimes alterations of things that affect transport or nuclear pore components tend to create aggregates in the neurons and in cells. Sure. And every time neurodegenerative diseases, and I'm not an expert on that, but if something that we have learned, everybody, is many of this Alzheimer and all these degenerative diseases, they have aggregates of things that are proteins that are not destroyed properly, they are misfolded, they accumulate, they precipitate, and they kind of create a problem in neurons, and they have this toxic effect on these neurons. So I know there's a lot of people working on that. For example, University of Pennsylvania, there's a group specifically working on how defects on the transport can affect neurodegeneration. Therapeutically, I don't know how this could be exploited unless there is something that you can push towards. If the problem is that cells cannot import things into the nucleus, if you can push the import somehow or block the export, because we had drugs either way. Some of the drugs we used in the past, we use them to tune down nuclear import, so we can inhibit nuclear import. But you can also inhibit nuclear export, so you can kind of change the balance of traffic in both directions, which at the end of the day, you need a steady state to things to work fine. The problem is when there is dysregulation of nuclear import and export. So I wonder if that could be a strategy for this kind of diseases. In some cases, many of them are associated to genetic changes. So then maybe we are adding there to a genetic component that is then difficult to treat, right? As I mentioned before, with the P53 in cancer, unless we find gene therapy that works, which when I was studying in college, that was like the big promise. When I started studying, like, oh, gene therapy is going to be, the future because we're going to cure the genes that are mutated. But that was in a long time ago. And we're still here thinking about gene therapy. There are some cases coming out of some pediatric, I think, leukemias and other that are starting to introduce some genetic changes in the cells and then try to utilize that. But it's not mainstream at all. So that would be the future for many of these alterations are actually mutations. Otherwise, we are forced to treat the function that we detect. So if it's a lack of function or something, how we can reverse that by pushing through with a drug in the opposite direction, right? Yeah. But yeah, the nuclear parts have been also detected in neurobiology and problems associated with neurodegeneration beyond cancer as well. Yeah.
Very interesting. Veronica, if you would, you're located down the street here, Sidney Kimmel Cancer Center, major center of excellence in cancer. But so I like to, you're in Philly, I'm in Philly. I like to profile sort of the Philly bio ecosystem. And as you were mentioning, there's other great organizations up the road. We have Fox Chase Cancer Center, Worcester Institute down the street here. Just say a couple of words about sort of the Philly sort of cancer bio ecosystem, if you would. It seems like maybe we don't get as much press as Silicon Valley or Boston or whatever, but a lot of exciting stuff going on in Philly, aren't there? On cancer.
Well, we may not have the whistles that other people may have in marketing, but there is a very big Philly-based cancer research, I would say even collaboration, but we have a very good multi-institutional efforts here, comparable to many other big city areas in terms of big university and institutes that are doing really great job, not only in cancer, in other specialties as well. But focusing on cancer, I mean, we have, as you mentioned, we have our cancer center, the Sidney Kimmel Cancer Center at Jefferson, Fox Chase, and Worcester, UPenn, and it's we have Drexel as well, which is part of the consortium here with Jefferson. We have many colleagues that actually we collaborate frequently. So it's a very powerful, local cancer enterprise, let's say, multi-institutional. And we actually reach out in our own interest, for example, in the study of GU oncology and prostate cancer. We have a multi-institutional working group that is like the Philadelphia Prostate Cancer Working Group that I have the honor to co-lead with one of my colleagues here, in which we have everybody from working on GU malignancies that is part of it. So we actually revamping it because we stopped it because of the pandemic, but now we are restarting again virtually in the fall, and we invite everyone to present. We have external speakers, we discuss science, we discuss new advancements in bladder and prostate cancer. And that is from different institutions, including UPenn. We start all the ones that I mentioned, Fox Chase and ourselves. So that speaks about that we have a mass, a critical mass. And I'm talking just about GU malignancies. So imagine everything else because we have other expertises as well, right? Sure. So there is a big mass of cancer research in here. And it may not be in the news every day, but there is, and good quality. So that Philadelphians should be proud.
All right, that's why I love to profile Philly when I can, and the fact that had the opportunity to talk to you and your thought leadership in this area just adds to it, because I think I think Philly's exciting.
And I guess that's why also there is a lot of biotech and pharma in the outskirts of surrounding us, because where there is knowledge, there is also companies that are close by. And then we have a big foundation star set here in Philly, right? Like the American Cancer Society, and we have the ACR offices right here around the corner. And I've been in both places, so it's when I wasn't here, I didn't realize that it was so close. I was living in New York. I never went to any AACR, ACS, headquarters to talk to people, and they are set here, right? So it's for a reason. So it's a big hub of cancer research, and that's for sure. Yeah.
Exciting times. Veronica, while I have you, just getting close to the wrap up here, but obviously you've not just obviously in Philly, but at your time at MSKCC and your work in Barcelona, you've had an opportunity to work with, meet many fascinating people across the bioscience ecosystem. I'm giving you the floor back. Take some time if you want to shout out to anybody in your current lab at Jefferson, other collaborators that are really very instrumental, not just in what you've been doing, but some of the programs moving forward.
Well, I have been lucky to work with great colleagues and mentors through my career, both when I was training in Barcelona. Obviously, I wouldn't be here if it wasn't because I had people who believed in me and invested in me and taught me to be a scientist, right? So I learned pieces from everybody, and that took me here. And it's funny because sometimes when you are on it, you don't realize, and then it comes to you later. It's like with your parents, right? 20 years later, so you appreciate what they did and why they did what they did, right? So with mentors, to me, it's kind of the same. Now I understand a lot of the things they did, and I wish I could talk to them now and say, wow, this was wonderful what you did here. And I understand why you did that, and that was really nice, and that stood to me, and I'm doing it the same way. So that's a common theme that I'm finding, that even with my more recent mentors, I understand many things and say, now I'm doing it the same way because I learned it from them, and it makes so much sense now than when I was a trainee, right? And that's one thing, the mentors that you have and the mentors that I had, I was lucky to learn from them. Either sometimes you learn techniques, sometimes you learn the style, the way of thinking, and the way they coordinate people. You learn little pieces from people, and that makes your own yourself and how you run your own group, which is what I'm doing now. And then it's the mentors that help you. Once you're a scientist, they're independent scientists, that they promote you, right? And then they put you in the stand and say, hey, look at what she's doing. Look what this group is doing. And obviously I have those in Sloan Kettering because I got an award there, because they recognize my work as a way of saying, look at this person, right? It's going to be doing interesting stuff. And then at Mount Sinai, I started my lab at Mount Sinai, so I got the first opportunity there. So I'm grateful for people believing in me and that I could have interesting research being done in a lab by myself. And then investors and foundations and NIH who believed in me. The Prostate Cancer Foundation were one of the first foundations who invested on our science, and sure after that the NIH funded my lab, and that's really, really, really important for us as investigators. It's taxpayer money. You're being trusted to develop research program and find answers in a hot topic, and that's very important, and that feels grateful at the same time of responsibility, but it's really invigorating. Like we got this, we're gonna do this. We got we got funding and the trust of the NCI, the National Cancer Institute, and then obviously people that gives you advice. And I have people here at Jefferson and my personal life, my partner, my husband. I mean, I'm a mother of 3, so I have 3 daughters, and I'm able to do what I do because I have the support of my partner, right? Otherwise, I know it's very challenging and for women in science to coordinate your personal life and run a lab. And I can do it. I'm like, I can do it because I have a person who understands that wants me to do what I'm doing because he believes in me and he doesn't think I have to compromise anything. We can have everything. You can have a happy family, but you can have your dream of being a scientist and also have a, generate knowledge and help the society. And so that I'm very grateful for that because I know it's not easy and I'm very grateful for that and people who believe in me. And the science we do, and we can do what we can do. So that comes from parents, friends, partner. My daughters are a great inspiration right now. People may think, oh, once you have kids, you are not competitive anymore. But I became so much efficient than when it was a postdoc, believe me, because I know the time I have.
Oh, yeah.
When you're training, you have the whole day and whole night. You can stay until 3 a.m. in the lab. You know, if something doesn't work, you repeat it. You don't have to rush. But now, hey, you have to do this in that time frame. And there are deadlines. I am much more efficient now than I was ever in my life, and more productive. So that's a message that I would like to send, that the productivity of mothers in science, it goes up. It doesn't go lower if you have the right support. So that's if you have the right support from your peers, colleagues, chairs, institution partners, you go up because you become very efficient in what you have to do, what you want to accomplish. And then for me, in my case, it's important to do that because I'm a better person at home because I'm doing what I like, and I'm very productive in the lab. So if I were home all day, I wouldn't be the best mom I can be. I'm the best mom I can be because I'm very productive in the lab. And when I'm home, I'm 100% there. So that's a message that I would like to convey. You can have everything. You can be a scientist. It's not easy. I'm not saying it's easy, but if you work hard and you have the proper support, you should go for it and follow your dreams, work hard. And you want to cure cancer, you have to think you want to cure cancer. You cannot say, that's impossible. I cannot do that. Or I want to have a family. I have to compromise. You don't have to compromise. You got to keep pushing. And that's how you make a difference. And I don't know if we're going to cure cancer, but we're going to try it. And in the meantime, I hope I can train people. I have a good group of, my lab is amazing. It's growing. I have 4 people now. Most of them are women. So I like for them to see that everything is possible. I'm not telling them that you have to have kids or not, but I want them to see what is possible without even saying it. So just live by example. So those are probably the main messages I would like to say, and that we are in this fight together with patient advocates, with foundations, with funding bodies, and we're going to tackle this. And it's difficult, it's complicated, it's hard, but there's a lot of people trying very hard, and we do try very hard to find answers and put together efforts to have more options for patients early in the process, not late.
Excellent message, and really, Veronica, wishing you sort of the best with all this. I mean, you have a lot on your plate, but you clearly have the energy and wherewithal to move all of this forward. And once again, just I want to thanks for taking the time to talk to us. For everybody that's going to be listening on the podcast or watching on the YouTube channel, you've been listening to Dr. Veronica RodriguezBravo, Assistant Professor, Department of Cancer Biology, Sidney Kimmel Cancer Center here at Jefferson University in Philadelphia, really working on the cutting edge of understanding the initiation, progression, and ultimately new therapeutic modalities for cancer. Veronica, once again, thank you for taking the time out of your schedule to do this. Thank you for everything that you're doing in your lab in the Philly bioscience ecosystem. And as we like to say here on our show, thank you for helping to create a better tomorrow through all your work. Really very fascinating stuff.
Thanks to you. It was a great pleasure, and I love this initiative you're taking, because it's very important that we can communicate all this to the rest of the world, in this case, the rest of Philadelphians, right? It's what we call outreach and communication of what we do. That's very important. We are all together in this, and we have to work both sides. So they understand, we understand, and we have a common goal.
Thanks so much.
Thank you very much.